Okay, my mic's on. I'm going to ask everybody to find your seats again so we can get started with the next session. Great. I'm Milan Mrksich, a faculty member here at Northwestern, member of the IIN community, and again, want to welcome everybody back. I unfortunately wasn't here this morning. Well, not unfortunately. I was teaching, and those of you who know our students here are just spectacular. They're really great to be in the classroom with, and we didn't want to skip it, Nicholas. So and these kids, I just share with you one story. One time I taught in the student evaluations at the end of the quarter, one of these students said, if I found out that I had a terminal disease and I had 2 weeks to live, I would want to spend every hour in Professor Mrksich's class. It was extremely touching until I read the next line, which said, because it would feel like a year. So anyhow, it's a pleasure now to introduce one of the world's and foremost leaders in molecular engineering and polymer science, Professor Tanja Weil. Professor Weil is the director of the Max Planck Institute in Mainz for polymer research, position she's held since 2017. Her research focuses on the design of interactive macromolecules and hybrid biomaterials with powerful applications in material science and nanomedicine. She's trained as a chemist at the University of Braunschweig and Bordeaux in France, and she earned her doctorate at the University of Mainz, where she really started beginning to develop her signature approach of combining chemical precision with biomedical purpose. She had an industry experience at the Merz Pharmaceuticals Company in Frankfurt, and faculty positions at the National University of Singapore and later Ulm University, where she directed the Institute for Organic Chemistry. Throughout her career, Professor Weil has pioneered systems that move beyond passive drug delivery. Her research bridges materials science, cell biology, and other fields to create active biomaterials that dynamically interact with living systems. She's developed materials that respond to disease markers, and when combined with molecular transport systems, can penetrate deep into cells, even into organelles, including mitochondria, to perform complex functions and address therapeutic targets. Her work addresses critical challenges in cancer, neurodegenerative diseases, and genetic disorders, demonstrating how advanced chemical designs can be harnessed to solve fundamental biological problems. In addition to her faculty contributions, Professor Weil plays an active role in shaping science policy and advancing research excellence. She serves on the Senate of the German Research Foundation, the Senate of the Leibniz Association, and has been a member of the Leibniz Evaluation Panel, really important roles in the German scientific ecosystem. She serves as an associate editor at JAX, a member of the editorial advisory board of ACS Nano, and really finds time to support our community in many other ways. She's been properly and well recognized for many contributions, the Otto Hahn Medal from the Max Planck Society, the prestigious ERC Synergy Grant from the European Research Council, and I'm going to save some time for you to talk about your science, but many others. And so let me just welcome you as a model for the type of interdisciplinary research that we really value and respect here at Northwestern. And please join me in welcoming Professor Weil to the podium.
Thank you. Yeah, many thanks, Milan, for the very nice introduction, and also many thanks to Chad for giving me the opportunity to speak here today. It's a great symposium, and I would also like to congratulate personally our award winners, who are really source of inspiration in our society. Yeah, so thanks for their great work. So today I will shift a little bit the focus towards nanomedicine and towards medicinal applications of soft matter. And I will try to explain to you how we merge soft matter and living matter and control cellular function. And we are looking into these strategies from a very fundamental perspective, but I will also show you some applications that we are heading for. So in order to give you a little bit of an introduction into the general field, I would also like to start a bit more general for you. So I would like to highlight how important living systems that are all around us, how important they are. So they are typically made out of cells, and I mean, they shape really how we live. They shape how I'm digesting, metabolizing, what I'm thinking in this moment. So this is all based on structures that are building up and that are being converted. And this is all very important in order to be able to adapt to an environment, to respond to stimuli, and to create energy, and so on. So if you take a look into the cell, so this is taken from the famous movie, The Inner Life of the Cell, but I really like it because it gives some kind of idea how molecules form supramolecular structures in nature, and how these structures are really building up by non-visible forces, and how these structures then guide further interactions with other molecules. And this is all something which is operates in a very concerted and autonomous fashion. So you have molecules that are binding at distinct positions, and you have dynamic processes going on. And this all later on tells us how a cell is viable and how a cell metabolizes and lives. And not all cells are the same, so there are also fundamental differences, and I will also come to that a little later. So the cell is a source of inspiration for us, and how the cell produces structures and also degrades structures is something we are also trying to emulate in our work. I would like to highlight also one very fundamental principle of life, and that is self-organization. Very precise self-organization from distinct molecules that then interact and form larger nanoscale structures, for example, here, protein fibrils, that then can form a network that is surrounding cells. And this network is also not dead. It's not dead matter, but it's active matter. So the soft matter structures around cells, for example, they are very important to keep structural integrity, but also for communication between cells. And this is also a source of inspiration for us because nature is actually capable of inducing changes in the cell by just rigidifying, for example, the matrix around cells. And so this is also something which I would like to highlight here. So the extracellular matrix, which we will also look into in a moment. Yeah, the extracellular matrix is a fibrillar network that's surrounding cells and that is actively engaging in many cellular processes. And it's actually also very important if it comes to, for example, tissue formation, healing, but also tumor progression. And having said that, I would like to move over to our own research and explain to you how we are integrating soft and living matter. So soft matter, synthetic macromolecules, or polymers, as shown here. So these are all these are all functionalities that we are, or architectures that we are also using in our group. So we are working with polymers. We are working with peptide nanofibrils, and I will also zoom in on my next slides. And also protein and peptide coacervates, vesicles, and also gel networks. And in order to merge them with biological matter, with living matter, you need to engineer the interface. And that is something which really interests us. And here I would also like to show you a few strategies. So how to combine soft and living matter, and how to engage really soft matter to control how living matter functions. This is one of our big ideas and also dreams. So my talk is separated, or it's divided into 3 topics. So first of all, I will talk about intracellular nanostructures. So how can we bring in small molecules into the cell and then control how they are assembling and forming an own fibrillar network inside the cell? The second part will be extracellular programming of cells. So here we are trying to control and also engage cells in introducing a new network around the cells and control cellular processes. And the last part is rather short, unfortunately, but it's an emerging part, also an emerging topic in my group, and that is to design soft matter and living matter that are co-evolving and also exchanging metabolic information. So let's start with the first part, and here our working horses are peptides, amphiphilic peptides, and for the non-chemists here, so these are basically small molecules, still relatively small molecules made up by amino acids that are linked to each other, and you can control really the linkage in such a way that you have a precise pattern of polar and unpolar functionalities that then can organize in water, for example, into these cross-beta sheet-rich, very ordered fibrillar nanostructures. And this is also shown here in the transmission electron microscopy images where you see at the nanometer level, so they have a thickness of about 4 to 5 nanometers, but then lengths can cover up to the macroscopic regime. So these nanostructures very much depend on the sequence of these peptides. And we are not only working with these two-dimensional peptide networks, but we are also using automated design and screening to identify entirely novel peptides that are forming fibrils. And a very new and recent example, and I hope you can see this here from the video, is a very thick fiber, which has about 20 nanometers in diameter, and again, very long, which is formed by a 9-mer peptide that forms these new honeycomb-type structures in the interior, so that you form channel-type architectures. Yeah? And this is something which we also find quite exciting. And these structures, they are not just two-dimensional networks, but they can also extend to the third dimension. So if you now introduce these peptides into aqueous environments, they will form fibers immediately. Yeah? And if you would like to bring them into a biological environment, such as a cell, you need to be able to control fiber formation. So the cell is actually full of nanofibers, yeah, and the inside also extending here with these protrusions to the outside. And so it's a relatively dense network. And we were wondering whether we can program at the molecular level molecules that are responding to biological stimuli. So if they are entering the cell and if they are then encountering different environments inside the cell, that they can convert chemically into different structures that at some point form a self-assembling structure that forms at the location where we would like them to form, where they would form also nanostructures. And I'm not going to show you all of the chemistry here, so don't be afraid. Yeah, we are synthetic chemists in my group, and we synthesize a lot, but I just would like to highlight the general principle. So, so this is a linear self-assembling peptide. So if this peptide is exposed to aqueous environment, it will form fibrils immediately. If you stabilize this peptide in an isopeptide, so in a kinked form, and attach a caging group that can respond to a certain stimulus, then this molecule is fully water-soluble. It will not aggregate. However, if it encounters a stimulus or two stimuli, then the caging group can be cleaved, and primary amine group is then formed that at physiological conditions would then show an ON-acyl transfer to this linear form that then forms this network. So here you see the soluble bioresponsive monomer, and here immediately a self-assembling monomer forming this network. So in principle, by just removing the protection group, you can control fibril formation. And this is something that we are trying to shape for certain environments in the cell. So we are looking at the cell more from a chemist's perspective. Yeah? So we are investigating and also trying actually to figure out by measurements inside the cell, so what kind of pH gradients do we find? Yeah, where do we have which pH? Where do we have which amount of or quantities of glutathione, so reducing agents inside the cell or oxidative conditions, different pH cycles. And then we are trying to design caging groups that respond to one or more of these stimuli. And this is just shown here again as an overview slide, so from work from Yong and Zara, so where we have again this linear self-assembling peptide that is then stabilized in this soluble caged form. And then upon the introduction of these caging groups, we can control whether uncaging occurs in the presence of glutathione, enzymes, light, reactive oxygen species, or pH. In particular, light is very useful from the outside to control when and where fibrils are forming. But the chemistry is quite complex, so I'm not going to dig into this now, but I will show you some illustrative examples. So with these caging groups, we can actually program when and also where molecules assemble inside the cell and analyze how the cell responds to it. So one example is shown here. So we have a sequence that allows cellular uptake, which is then combined with this lock, this caging group that responds to glutathione in the cell, and then the pro-assembling sequence that forms the nanofiber. And this is a lock that responds to glutathione in the reduced form that then unlocks this molecule once it enters the cytoplasm. So it's a relatively simple lock. And now if you look into the cell, and here we have a cancer cell, a triple-negative metastatic breast cancer cell that is not responding to chemotherapy anymore, and you can see on the left-hand side, so it will start in a moment again from the scratch, so that already after 15 minutes, you see the formation of these fibrillar architectures, and there's a very strong impact in cell morphology. So you see here after 10 minutes, yeah, how these aggregates, and from these aggregates, these fibrillar structures are starting. And if you zoom into the mitochondrial network, you can also see how these nanostructures are affecting, at a high dose, though, the mitochondrial network. And then if you look into energy metabolism, and this is complex, and the slide is a bit too dense, but I just want to say, so we take methods that biologists develop where you can measure on the cell the oxygen consumption rate, so how well a cell can breathe. And then we add our sample and investigate, and actually what we can see is that the capacity to breathe, so that means also to metabolize, to perform glycolysis, for example, inside in the cytoplasm, or oxidative phosphorylation in mitochondria, so that this drops immediately, also very, very quickly after the addition of these compounds. And there's a concentration-dependent toxicity that goes down to 0. And we have comparisons also in this work where we use classical chemotherapeutics against which this cell line is not is resistant or has developed resistance, and we could see that really here we are killing all the cells which form, so when we form these nanostructures inside. If you look into 3D models, so in my group, we are also growing tumors in 3D. We are using patient tumors, but we are also growing so easy tumor spheroids out of one cell type. And then you can see these isopeptides in a three-dimensional tumor, again, of the triple-negative breast cancer cell line, the MDA cell line. And the peptides are linked with a chromophore, so you can see the emission here. You can see basically also that the cells are entering into cell death. So this is something which we can stain and also visualize. And if you do slices of the tumor, so you cut thin slices, you can see that these small molecules, they can penetrate deep into the tumor. And this is also something which is visible here. So not only at the surface, but also deep inside the tumor. And we see already after the first application quite a decrease in tumor mass. So we are quite quite happy about these results and now also pushing forward towards in vivo experiments. Let me move one step further. So with these nanostructures in cells is relatively, let's say, also straightforward to kill cells. But we were also wondering whether we can ramp up new functions of a cell. So not only induce cell death, but maybe implement new features. And here we are particularly interested in certain cells that play an important role in the tumor microenvironment, and these are cytotoxic T cells, yeah, that are also important for CAR T cell therapy, but I'm coming to that in a moment. And these cytotoxic T cells, they have an inactive and an active state. And the tumor microenvironment, because of its acidity, often deactivates these cells. And then they are often called exhausted, and they are not able to attack tumor cells anymore in a very simplistic, in very simplistic terms. So we were wondering whether we might be able to stabilize or to introduce fibrils that somehow stabilize these cells and make them a bit more active and robust. And again, the same molecule as I've shown has been applied here by Sarah and immunologist Tobias Bob. And so if you incubate cytotoxic T cells that have been also received from donor patients, then you can form these nanostructures inside these cells, and we do that at very low concentration of only 5 micromolar. And what happens then is that the morphology of these T cells actually changes quite drastically. So you can see here a treated T cell, so it becomes more round-shaped. And if you investigate how these T cells then behave in a real-time deformation cytometry assay, yeah, where cells are basically forced to move through a small channel, then untreated T cells, they respond to shear forces, whereas our treated T cells, they are not responding to these shear forces anymore. We have an altered morphology, interestingly, increased cell stiffness, reduced deformability, we did many tests here, and an increased elastic modulus, which we found interesting. Yeah? And then we were wondering, so what happens here? So why do these cells show such a behavior? And nowadays, you can quite easily look into the proteome of cells. And we did a proteome profiler analysis and really found out that there are many different protein states changing inside the cell after treatment. So on the one hand side, amino acid synthesis is really ramped up dramatically. And we also see that phosphorylation of certain proteins is increasing, and that pathways that are connected with survival, activation, effector function, metabolism, and survival, so that they are upregulated. Yeah, and this is nothing we could, we could engineer from the scratch. So this was something which came out of just curiosity and some investigation where we did not know at the beginning, yeah, how this would turn out. So, so that was interesting because our immunologist said, oh, you are producing cells that are fitter. Yeah? So you are increasing fitness of cytotoxic T cells, which could be quite interesting. So what we then did was we used these fitter cytotoxic T cells and incubated them with a specific T cell engager that is binding to HER2. HER2 is overexpressed also in certain breast cancers as one target receptor, which is also used for patient treatment, and anti-CD3 at the surface, so that we can actually combine our treated cytotoxic T cells then with the cancer cells and investigate the cytotoxicity. And what we found out was that after treatment, that the cells were actually tenfold more active. Yeah? So you could dose much lower in order to have an cytotoxic effect here, which maybe opens new opportunities. Yeah? So because peptides are simple, they are easy, they can be applied easily to enhance the activity and maybe the robustness in vivo. So we are still waiting for the animal experiments and for the allowance to do that. But that would be actually, that would, to my mind, be quite exciting. So yeah, as I said, CAR T-cell therapy, that's something which is also important in minds. We have companies like BioNTech going in this direction, and this is where we hope that we can, with our strategy, then convert maybe an exhausted T-cell or a T-cell that is easy to exhaust into an active T-cell, and later on, yeah, move this forward and use it hopefully also in patients. So with this, I would like to take my last 10 minutes and switch gears and talk now about the extracellular matrix and how the extracellular matrix can also program cellular fate. And here again, I would like to highlight that the cell has many receptors on its surface. So if you engage a soft material with a cell, then likely the viscosity and the structure of the soft matrix around will also affect cellular function. And we were wondering, again, here also, whether we can change and alter protein expression just from the outside. And so one rational approach where we were extremely pleased to see that it worked out was that we said, okay, first of all, can we actually slow down assembly and give the cell time to recognize that there is a self-assembling aggregating molecule inside the cell to spill it out and form a matrix? So the idea is, because it's not so easy to prepare a matrix in the surrounding of the cell without killing the cells. So we thought maybe we can engage cells to do so. So we have, so this is a fast assembling monomer that you've just seen, yeah, that assembles extremely quickly. So the cell cannot respond. Yeah, so in cancer cells within 15 minutes, we saw a cell death. We designed a sequence that shows slow assembly over hours. And what happens then is that fast assembly, so here, this was what I've just shown. Yeah? So you form these aggregates inside cells and within 15 minutes actually introduce apoptosis. Whereas in the slow assembly, the cell has time to react towards the aggregation of these molecules, and it's forming an own matrix around the cells. Yeah? And this matrix is not cytotoxic, but gives us an opportunity to further actually explore how the density of a matrix affects proliferation and maybe also to chemically modify the extracellular matrix. So this is a picture how this matrix looked like here in microscopy, and you see in green these fibrillar structures, and you see also cellular protrusions actively engaging with these structures. And here's an endocytotic vesicle that actually expels peptide aggregates to the outside where this matrix forms. And then, so you can introduce easily, for example, azido groups. And azido groups are very important for the chemist because azido groups give you the opportunity to do chemistry. And you can hook, for example, labels there where this matrix forms. But also then, depending on the density of the matrix, if you boost the density, then at some point also protein expression changes. And we are ramping up, for example, endostatin as an anti-inflammatory or a protein linked to anti-inflammatory pathways, and we are decreasing, for example, proteins that are linked with pro-inflammatory pathways. Yeah, and we actually we thought that this is a nice way to form a homogeneous environment around cells that is functional and that also offers you to further customize this shell. In my next example, I would like to show you how we can then now train the cell to produce such a matrix on its own based on a conversion of a metabolite of a cell towards an active ingredient that actually produces this network from the outside. And here, I would just like to highlight Otto Warburg, who received the Nobel Prize, and he looked into the altered metabolism of many cancer cells that are ramping up glycolysis inside the cytoplasm, and then as metabolites, lactate is increased. And lactate is not only a metabolite, but it plays a very important role also as a signaling molecule in immune suppression, metastatic dissemination therapy resistance, and in particular also the acidity of the tumor microenvironment, which is also linked to T-cell exhaustion. So we thought, what happens if we now take this metabolite, and if we convert this metabolite via standard chemistry, actually, locally, so here's lactate, and if we would be able to convert it into hydrogen peroxide locally, and then if we make use of a cross-linking chemistry that nature uses, yeah? So Nathan knows this chemistry actually very well, and so muscles actually use this chemistry to connect to stones, or we use it also for pigment formation in our body. So, but this cross-linking mechanism allows you to form a network, a gel network. And we were wondering what happens if we take a polymer with tyrosine groups, so with OH groups, and we produce locally hydrogen peroxide, so whether we could form a gel around cells. And this is the polymer we are using. We are using modified dextrane, quite simple, and then two enzymes, horseradish peroxidase and lactate oxidase. And first of all, I would like to show you that we are actually, so normally cancer cells are ramping up lactate, and in the presence of our polymer with these enzymes, the lactate level even decreases. So lactate is used as a fuel and it's metabolized, and a gel is formed with increasing stiffness over days. And this is how this looks like. So you add the polymer to the cancer cells together with these enzymes, and then immediately, and we tested this for many different cell types now, immediately a shell is formed around the inner tumor spheroids that you can see here. And very interestingly, this shell, so the cells cannot escape this shell because it's a very dense shell. And so the cells are responding to this shell. So they are upregulating proteases to remodel the matrix around, but this is not possible because it's a chemically linked, it's a CC bonded network. Yeah, so it's amazing to see how many defense mechanisms such a tumor has. So the tumor is trying to affect matrix stiffness. The tumor cells really try to move outside the gel, which is very difficult because of this highly cross-linked network. And they are trying to recruit immune cells, which is also not possible. And what we can see is so many different proteins are up or down regulated, but in consequence, the tumor is not growing anymore. And cells are, so the tumor basically remains like within a cage. And this is, to my mind, quite a nice opportunity because this approach is specific for metabolic state, yeah? The state when lactate is produced. If lactate is not produced, this scenario wouldn't happen. So you really tailor your chemistry to a certain state of a cell where other cells would not react. Now I have an option to finish now and move over. I don't know, or do I still have 2 minutes? Most likely it's finished, right? 2? Yeah. Then I would just like to show you very briefly, because here we used a similar mechanism. So we also used the lactic acid encapsulated within an artificial cell and said, okay, now can we co-evolve a system together with cancer cells and offer a catalytic cascade, so like an actuator where lactic acid is catalyzed into pyruvate, which is then catalyzed into acetaldehyde. And acetaldehyde is a byproduct if you consume alcohol. So if you produce artificial cells, which are these vesicle-like systems of dimensions of cells with these enzymes in the interior that cannot escape, and you can encapsulate 2 different enzymes in, then you form something which we call a metabolic actuator. And this metabolic actuator is now able to pick up lactate and convert lactate into acetaldehyde that then diffuses out and then affects cell viability. So it's a system where you have an input signal, a processor, these are these 2 enzymes, an interface, and an output, which is cell death. And here I would just like to show you actually how this looks like in a 3D tumor. So you can grow these cells together. Maybe it's better to see here. So you can grow these artificial cells together with a three-dimensional tumor. And indeed, lactic acid in this three-dimensional tumor model is depleted. Acetaldehyde is increased. And one can clearly see how tumor spheroid growth is stopped, and toxicity actually is more pronounced in comparison to the control cells. So this is something where we are offering, hopefully for the future, systems that are not a single shot, but that remain sustained in a biological environment and respond to a certain cellular state, to a metabolic state, and where hopefully at the end a single dose, so a single entry, so a single application of these systems would allow to, for example, stop growth of a tumor. I mean, this is something which is far-fetched, but definitely one of our goals. So here are, here's my group. I don't know why Celine is missing, but Celine and David did also most of the work together with me, and here our funding agencies, and the synthetic cell has been done together with Stephen Mann from Bristol. Yeah, thanks a lot for your kind attention. I'm happy to take your questions. Thank you.
Okay, time for questions, and if you stand up, a microphone will appear. Oh, one back here in the aisle.
Thank you for the great talk. I was curious about the peptides that you were talking about in the beginning for rewriting intracellular processes, and I was wondering how they were uptaken by the cells and how you combat the endosomal escape process.
Actually, that's a very important question. So cell uptake is also not always so easy, and it defines via which pathway the molecule will also enter the cells, which is also very important if one considers the compartments this molecule will experience on its way. So here I basically showed you a relatively easy sequence. So it's a TAT sequence, which is taken from HIV. And it's a polycationic sequence that induces endocytotic uptake via endocytotic vesicles. But you can also add, for example, targeting groups. So this makes the molecule a little more complicated. So for example, somatostatin receptor ligands that are binding to somatostatin receptors. So here you can, in principle, also implement some kind of cell type selectivity. But this is not our major goal. So we only want to get into the cell, but then the specificity we would like to achieve is based on the cellular state and the cellular metabolism and the conditions that the molecule will experience, either in the microenvironment or inside cells. But targeting can be implemented also.
Thank you.
In the front here.
I guess it is on, yes. I saw your slides with the tumor cells.
Yep.
What happens with regular cells? Do these kill the regular cells?
Yeah, so in this example where we applied a lot of these small molecules, that would also kill regular cells. So that was, in principle, the first example where we simply tried out what happens if we bring in, I mean, we are talking about 50 micromolar, so massive amounts of molecules into the cells. But now we have systems like so that we can dose lower, like for the cytotoxic T cells. And there the cells are very happily alive. They're even more robust. So this is all a matter of how you tune also the chemistry here. So it must not necessarily be toxic for healthy cells. Actually, that's one of the ways what we try to achieve with the chemistry, that we have a selectivity towards several stimuli that you find in cancer cells and not only one.
Thank you.
In the back, yes.
So I saw heptamer at the beginning of your presentation. Is the length of the peptide is important for cellular uptake?
Sorry, where are you?
Oh, sorry, in the back here, standing.
Okay.
Right here.
Sorry, I didn't see you. Would you be able to pardon me?
Is the length of the peptide important?
The what?
Length of the peptide.
The length of the peptide, yeah.
Yes.
The length of the peptide is, so it defines often the speed of assembly. And the speed of assembly is important. That's, so you have a number of variables, actually. So in the slow assembly that I showed, it is a longer peptide. So we wanted to slow down the assembly kinetics. And if you slow down the assembly kinetics, also fibrils occur, but the cell, for example, has time to react and to repackage them. And then this extracellular matrix forms. Yeah, so the length can affect the speed of assembly. Must not, but it can.
These are normal peptides or modified peptides?
So these are, so often we have a chromophore attached, yeah, that we can follow, or phaser film dye that we can quantify assembly inside cells. But you can also do that fully with just native peptide sequences. Yeah? But so for us, it's very important to understand the mechanism. And in order to understand also the mechanism and, for example, kinetics, the dynamics, also whether they are, so where do they form? So that's something we are doing by adding or co-polymerizing, for example, with a dye that shows changes in the fluorescent lifetime. And then you can follow the assembly process from monomers over intermediates to the mature fibers inside cells.
So finally, how did you control the precipitation? You know, most of the peptides, they are,
How do I control what?
Precipitation.
Precipitation, you mean the liquid-solid transition, yeah.
Yes.
So this is something which, so if you form the monomers, yeah, so before you have a soluble system, but after the chemical reaction, you form the monomers, and then if you have a critical concentration, it's all dependent on the critical aggregation concentration, yeah, which depends on the sequence. Yeah, and then if you are above the critical aggregation concentration, then fibril formation will start. And fibril formation is typically relatively fast, unless you slow it down. Yeah, and so that means in the environment, so we have one peptide, for example, that is very, very fast and that forms fibers at the interface of the endosomal vesicle to the cytoplasm. And then you can really see a corona of fibrils forming around endosomal vesicles. Yeah, and that was a very fast one, for example, but with a different switch.
Great, and we've got time for one more, and it'll be back on the right.
That was a very nice talk, especially the extracellular peptide matrix, which keeps the cells functional. That is very impressive. But I do have a question about, clearly the cells are functional because the data shows so. I want to know, what are you tuning in the mineralization process or in the matrix formation process to make sure all the receptors are still open? And how are you confirming that the receptors are all open and functioning?
Yeah. So so that's actually one of the big problems when you try to engineer a new extracellular matrix around cells. Yeah. So in theory, you could also do it by just assembling something at the cell surface. But if you would do that, for example, with these peptides, then, I mean, we also did that as a control. You just get a chunk of aggregates, yeah? So not something that integrates into the matrix. And for that, this pathway from the inside to the outside is very important, yeah? Because then you are forming these long fibrillar aggregates, which actively, so where the cell is actively engaging with these fibrillar aggregates via filopodia, yeah? So this is something which we have also tested. And so what we see is that at low concentrations, I mean, everything, if you massively dose there, yeah, you would also affect proliferation. But at low concentrations, so far, we have not seen negative effects on proliferation, nor on protein expression. Yeah, but it gives you an opportunity to label and to do chemistry and, for example, introduce enzymes for new catalytic reactions in the extracellular matrix and things like this.
Okay, this has been a fantastic lecture, a great Q&A. Let's thank Professor Weil again. Thank you. Okay, great. It's next an honor to introduce our last speaker before lunch, but exciting because this is the award winner for the 2025 Kabiller Rising Star Award, Professor Michael Mitchell from the University of Pennsylvania, where he's in the bioengineering department. Professor Mitchell is recognized for his groundbreaking contributions to drug delivery and gene therapy, particularly in the design of lipid nanoparticles and those platforms for targeted delivery of mRNA to cells and tissues and organs with real specificity, really directing which tissues these genes get into. His work has enabled precise delivery to complex biological targets, such as liver, heart, brain, placenta, fetal tissues, and immune cells. He's expanded with his work greatly the therapeutic potential of the RNA technologies. And these innovations are driving new treatment strategies in cancer, autoimmunity, and cardiovascular disease. A notable achievement from Professor Mitchell's lab is the development of a biodegradable polymer, suit of armor for CAR T cells. This is an approach that reduces inflammatory side effects and allows these cells to gradually recover their full therapeutic activity. This work represents a critical step towards improving the safety and durability of cell-based immunotherapies. And in addition to his academic achievements, Professor Mitchell is the co-founder and serves on the scientific advisory boards of a number of biotechnology companies, which are advancing non-viral delivery systems. These include Liberate Bio and Capstan Therapeutics. And I think the translation of his work is one of the areas that's really now rising and I think is going to really deliver a lot of significant impact from what's happening in his lab. Professor Mitchell received his PhD in bioengineering from Cornell University, trained with Michael King, and then was a postdoctoral fellow at MIT with Bob Langer. And his team at the University of Pennsylvania is a significant and multidisciplinary team that's carrying out the work that he'll share with us in a moment. He's won a number of awards, the NSF Career, Burroughs Wellcome Fund Career Award, and NIH Director's New Innovator Award. And you know, I think it's also, again, fair to say that, and the theme of the IIN and what we're so excited about, this is another beautiful example of a program that combines engineering and biology and materials and chemistry to really tackle challenging problems, and we'll see here momentarily is really exciting progress towards those. So let's please welcome Professor Mitchell.
Great. Well, thank you, Milan, so much for the kind introduction. And a huge thank you again to Chad Merkin and David Kabiller for the amazing time last night for the awards ceremony, and then the whole IIN for an amazing symposium today. So I'm really excited to share some of our work today. I run a biomaterials and drug delivery lab at the University of Pennsylvania, and a major thrust of our lab is developing next generation lipid nanoparticle technologies, which many of you are now familiar with due to the development of the COVID-19 mRNA vaccines. So as a bioengineer, I really love these technologies because they're an example of an interdisciplinary science feed. They're part messenger RNA technology that encodes for the antigen of interest, but also equally part nanotechnology. Unlike most drug delivery systems, when we think about the delivery system improving, making it safer or more potent in the body, in the case of the RNA vaccines, the RNA simply doesn't work without the nanotechnology. So it's equal parts nano and RNA to create an effective mRNA vaccine. And we now have many of these on the market. Of course, we have the COVID-19 mRNA vaccines. In the coming years, we'll hopefully have cancer vaccines based on mRNA as well. And this brought many different people to develop, together to develop these technologies. Our collaborators and friends, Drew Weissman and Katie Carrico at Penn, recently won the Nobel Prize for the nucleoside-modified mRNAs. Peter Cullis at the University of British Columbia developed the ionizable lipids for the LNP delivery systems, but many people contributed to this field of non-viral delivery to get it to the point where it was today, such as my former advisor, Bob Langer, of course, Chad Merkin, and we'll hear more from Nicholas Pepis later today, and then many others at the NIH, BioNTech, Pfizer, and Moderna that developed and scaled up these vaccines at an unprecedented speed. And now that we have this amazing example of nanotechnology where it's been dosed in billions of patients and it's been shown to be safe and effective, one of the major questions we have as a field is, can mRNA lipid nanoparticle technology broadly revolutionize pharmaceuticals? Can we go beyond the COVID-19 vaccines for new flu vaccines, for new personalized cancer vaccines, but can we go even a step further and use mRNA LNP technology to secrete and generate any therapeutic protein in the body? Can we use it to edit out deadly mutations that arise after birth? And there's certainly the potential to do so, but one of the key bottlenecks that we face in the field is the delivery issue. If we think about delivering the vaccine, it's a low-hanging fruit in that we get an injection into our muscle, it drains directly to the lymph nodes, but it's much harder to reach cardiovascular tissue, cross the blood-brain barrier, or penetrate deeply into solid tumors. So to do that, we really need new forms of nanotechnology to reach these difficult-to-target cells and tissues, and that's what my lab works on at the University of Pennsylvania. We're developing these next-generation lipid nanoparticle technologies as well as polymers that could deliver mRNA, but also a range of nucleic acids, DNA, circular RNA, siRNA, into target cells and tissues. We primarily think about this in vivo, but we also do a lot of work for ex vivo immune and stem cell therapy at Penn, and we even think about this in utero. So if a mother gets an in utero diagnosis of a deadly genetic mutation, can we potentially intervene shortly before birth with a CRISPR-Cas9 lipid nanoparticle approach to edit out deadly mutations so the baby can be healthy? When I started my lab at University of Pennsylvania, we primarily worked on this when I started in 2018, this concept of in situ CAR T-cell engineering. So CAR T-cell engineering is an ex vivo manufactured cell therapy, but if you could generate the CAR T-cells directly in the body using nanotechnology, you could really broaden the amount of patients that we could reach across the world that don't have the same standard of care. So I'll talk a little bit about that. This is moving into clinical trials, but since then we've been developing LNPs for a range of cell and tissue targets. I won't be able to talk about them all today. I'm happy to talk at the lunch or the poster session, but I'll specifically focus on our work in treating solid tumors and then some of our maternal health applications as well. So how do we think about this as engineers and nanotechnologists? When we design the lipid nanoparticle delivery system, it needs to satisfy numerous engineering criteria to reach the cell or tissue target. In one aspect, we think about the lipid nanoparticle as a protective carrier of fragile mRNA cargo. So the LNP that was developed for the vaccines is not one size fit all. We need to think about the cell target, the tissue target, but then also how it's administered in the body. So if we deliver lipid nanoparticles via systemic administration directly into the bloodstream, if we have naked RNA without the LNP, it's going to get phagocytosed by your innate immune cells, macrophages, neutrophils right away, or get degraded by serum endonucleases within the bloodstream, essentially recognized as a virus. But if we encapsulate it in these particles, we could evade innate immune recognition and serum endonucleases. They'll then circulate through the bloodstream. They then need to cross endothelial barriers and a negatively charged, dense extracellular matrix, and then they ultimately need to reach the cell type of interest for intracellular delivery. So if we have RNA by itself, it's hydrophilic, it's negatively charged, it's not going to be able to efficiently cross cellular targets by itself, but if we could condense essentially what we could think of as these long spaghetti strands into these 100 nanometer particles, they could then undergo endocytosis in target cells of interest. And then once we get into the target cell of interest, endosomal escape remains the major challenge in the field. It's estimated that once they get into these cellular compartments, only about 2 to 5% of nanoparticles escape the endosomes. So we design next generation materials, LNPs, that are ionizable in nature. So by ionizable, we mean that when they're at neutral body pH in the circulation, they're very safe, they're not toxic. But once they get into these acidic endosomes, endosomes where you have this low endosomal pH, these particles then become highly charged, so we're essentially flipping a switch. They're becoming positively charged, and then they could escape the endosomes to release the RNA cargo. And then lastly, we design it so essentially the LNP falls apart. We don't want it bound too tightly. We ultimately want the RNA release so it can undergo cytosolic delivery and then be translated into proteins. Or in the case of gene editing, we could translate it into a Cas9 protein, which can then go into the nucleus of cells. So to do this as a lab, we're developing an LNP toolbox for nucleic acid delivery. So I run a lab that's highly interdisciplinary. We have chemists, material scientists, and biologists working together to develop these technologies. A major thrust of the lab is novel combinatorial chemistry approaches to develop large libraries of what we call these lipid-like materials that we can make into LNPs. So as I mentioned, these are ionizable materials. They're neutral at body pH for safety purposes, but they become charged when we formulate them into LNPs and also when they undergo endosomal escape in cells. And to make these large libraries, we take a very simple 1 to 2 step synthesis approach. This is typically a Michael addition reaction or an SN2 reaction that we utilize, where we take different types of amines. So these amines shown here in red are ionizable in nature, and we look at a range of chemically diverse amines because different types of amines will transfect different cells or tissues better, but we can't make lipid nanoparticles with just these amines alone. So through these 1 or 2 step reactions, we could react different types of acrylate, acrylamide, or epoxide terminated tails. So these are hydrophobic, greasy tails, and make these materials into lipids. And as a result, we've developed thousands of ionizable lipids in the lab that are able to target different cells and tissues in the body and have unique endosomal escape properties in different cells. So this ionizable lipid is a very important component of the LNP, but it's ultimately one of 4 components. To make these LNP structures, we'll combine the ionizable lipid with what is known as a helper lipid. This is typically either zwitterionic lipid that can help encapsulate large RNA payloads. We'll combine this with cholesterol that gives the LNP membrane stability, and then a lipid polymer construct where the lipid is inserted into the LNP membrane, and then we have the hydrophilic polyethylene glycol polymer that gives your lipid nanoparticle a water-like coating. It makes it stable in solution for transport and shelf life, but it also gives it enhanced biodistribution properties in the body. So these are all combined in an ethanol phase. We then have our RNA cargo that's in an acidic buffer, and then through custom designed microfluidic devices in the lab, we can combine these 2 phases together through chaotic mixing, and essentially what happens is the acidic buffer will then come into contact with the ionizable lipid, and then through charge-based interactions and also hydrophobic interactions, you can assemble LNPs that are around 100 nanometers in size. We could do this at a small scale in the lab for cell studies and for mouse studies. We're also developing with collaborators at Penn microfluidic devices that are about the size of a quarter, and they could have anywhere from 250 to thousands of channels mixing and formulating LNP simultaneously. We could do this for large studies, for human studies, for NHP studies, but then we could also do these to make large libraries on a chip. So we can now make hundreds of different LNPs within a single microfluidic chip. We then also combine this with molecular barcoding technologies as well. And the reason we do this is when you make these very large libraries of nanoparticles, the challenge then becomes how do you evaluate them in an efficient manner in vivo. So very similar to a barcode that you'll have on a cereal box, we'll have LNPs that through nucleic acid barcoding, they'll each have a barcoded nucleic acid in them. And what that allows us to do is now we could take many different formulations that we normally evaluate one at a time in a mouse. You can now pool those together, administer them into mice, and then isolate out the cell or tissue of interest. And then through next generation sequencing, we could identify for a given cell target, for a tumor cell, for a neuron, for a stem cell, we could identify out of a pool of particles what is the best one that can reach that cell or tissue. So we combine these approaches to accelerate the design and identification of next generation LNPs to unlock delivery to cells and tissues beyond vaccines. As I mentioned, we work on a variety of applications in the lab. One I'll focus in on today is next generation LNP technology for precision cancer immunotherapy. Many of you may have heard of this technology that's now in the pipeline, the concept of a personalized mRNA LNP cancer vaccine. There was a nice article recently by my former advisor, my postdoc advisor, Bob Langer, who is one of the founders of Moderna, who discussed the development of these personalized cancer vaccines. And essentially what happens, how we develop these personalized cancer vaccines, is that a patient's tumor will get biopsied. We could then identify from those biopsies neoantigens that are expressed as tumors that could be targets for cancer vaccination. And when we identify those neoantigens, we can essentially make mRNA constructs encoding for those antigens that could be encapsulated into LNPs, administered to patients, and then these cancer vaccines can generate an immune response against the tumor. These are progressing incredibly well in clinical trials. They'll hopefully be on the market soon, but they'll work for certain types of cancers. Other types of cancers remain challenging to treat with these personalized cancer vaccines. Some of the challenges that we face is antigen identification. So what are these antigens, or what are the cocktail of antigens? And even if we do identify the antigen of interest, well, what if it's expressed in healthy tissue, and now we've created a vaccine that will target the tumor and the healthy tissue? And then sometimes, if there's several antigens that need to be encapsulated in an LNP, it becomes difficult to deliver these larger payloads. So what we're developing as an alternative in the lab, this is led by a very talented postdoc in the lab, Changcheng Shi, is we're developing a cancer vaccine that we could create directly inside the body without knowing what that antigen is. We call this concept in situ cancer vaccination, or creating the vaccine directly in the body. And essentially what we're doing is we're developing an mRNA lipid nanoparticle technology that essentially acts like a sledgehammer when it comes into contact with the solid tumor. So when we deliver this LNP technology, what happens is when it gets into the tumor, it'll lyse the cells, the tumor cells that are present within that microenvironment extensively. And when those tumor cells get lysed, they'll release that antigen that can be used to create a vaccine directly in the body. The antigen-presenting cells will then take up that antigen, traffic to lymph nodes, and then train tumor-specific T cells that can eradicate that tumor. And we call this new technology that we're developing, this is unpublished work, we call this a prodrug lipid nanoparticle technology, or a PLNP technology, that we're developing for synergistic cancer immunotherapy. I won't have time to get into all of the chemistry behind this next generation library of lipids that we're developing, but essentially you can see two parts of this lipid. We have the part shown here in black and the part shown here in red. The part in black is your traditional ionizable lipid. It has an ionizable amine head, it has tails, it acts as a delivery system, but then through linkage showed here with this disulfide linker, we can develop lipids that now have different types of drugs, prodrugs, that are attached to them. And in this case, we're delivering a drug known as an IDO inhibitor. What IDO is it's a molecule that's overexpressed in solid tumors, and we heard a great talk from Tanja just before. When you have high levels of IDO in solid tumors, it exhausts the T cells. The T cells become less prone to generate an immunotherapy. So what we're doing with this lipid nanoparticle approach is we're delivering a cytokine, in this case, into the tumor microenvironment. This is known as IL-12. It's a very potent anti-tumor cytokine. It could also generate immunological memory, so essentially the immune system can remember the tumor if it comes back. And we're also delivering this ionizable lipid that will deliver the IDO inhibitor, and it will prevent the T cells from becoming exhausted in this case. So we're developing this as a form of a synergistic cancer immunotherapy, both activating the T cells and also suppressing their exhaustion. I love showing this slide because this really shows the importance of developing next generation libraries of lipids and why they're not one size fit all. When we started this project, we screened a variety of industry standard lipids, so Alnylam's MC3 lipid, and then also the lipids by Pfizer, BioNTech, and Moderna. Basically what we could see, these are tumor cells that we're looking at here. When you see this signal in yellow in tumor cells, this means that the LNP is trapped in the endolysosomal compartments. lipids. It cannot release the RNA, but through generating next-generation lipids, these prodrug lipids, we can now have lipids that when they enter the cell, we get this green signal. This indicates that the LNP is getting into the tumor cell, but it's also getting out of the endosomes and subsequently releasing its RNA cargo to create a potent immunotherapy. So we then asked the question, in a proof of concept mouse study, can we develop this next generation technology to deliver IL-12 mRNA, reduce T cell exhaustion, and eradicate MC38 tumors? So these are a mouse model of colon cancer that we inoculate within these mice, and then we give the mice one of three treatments. One is the prodrug lipid by itself, no mRNA drug. One is the mRNA drug, but without the prodrug component, so a standard LNP. And then in red shown here, we combine the mRNA drug with the prodrug lipid component. And the results are quite striking here in this mouse model. So if we look here in green, if we just deliver the prodrug by itself, these tumors grow. If we look at the survival curves, these mice die after 50 days, very similar to the control mice that have tumors. If we deliver the RNA drug without the prodrug component, we're able to reduce tumor growth somewhat, and about half the mice survive about after a 50-day period. But if we combine the approaches where we deliver the IL-12 mRNA drug and suppress T cell exhaustion with the prodrug, we saw a striking result where we had 100% survival of these mice and complete remission of the tumors in this mouse model. So we had complete remission, and at this point there was no tumor left to analyze. So we asked the question, well, we can't analyze the tumor because it's been eradicated, but can we look into the spleen and see, is there a presence of memory T cells that have been generated that can remember the tumor, and if it comes back, if we get recurrence, can that immune system alone eradicate the tumor? So we did a subsequent experiment where we took the mice that had complete remission of the tumor, and then we take those mice and we re-inject a tumor essentially anywhere we can in the body and ask the question, without the LNP drug, has the immune system remembered that tumor and can now eradicate it? And we saw this very exciting result here. So we took the mice with the complete remission, we re-inoculate them in the tumor. The treatment groups are shown here in red. We see a little bit of a tumor growing at about day 6, but then what you see after that is that the immune system recognizes the presence of that tumor within the body, and then we can eradicate the remnants of that tumor and achieve 100% cures in these mice. Showing with this next generation LNP that acts as a delivery system but also as a prodrug, we could potentially create a vaccine now directly in the body against tumors. So this is an exciting technology that we're developing. Some of the challenges we face is that this is an intratumoral approach. This will work well for certain tumors, but systemic delivery remains the challenge for delivery into most tumors. And we're also doing work in this space. I love showing this image here. So many of you recognize this. For the very young kids, this was a movie called Star Wars. It came out in the 1970s. What we're looking at here is the Death Star, and we see the rebel forces are trying to get to the Death Star. But as you can see, there's interference. It's not easy to get to the Death Star. We have ships that are coming out of the Death Star to protect it. And the reason why I bring this up as an analogy is we're now looking at the role of, very similar to the ships coming out of a Death Star, we're looking at tumors that secrete large amounts of what are called extracellular vesicles. So these are almost like nanoparticles, naturally derived nanoparticles that are secreted by tumors, and we're asking the question, do these protect solid tumors from nanoparticle delivery? Because there's now evidence in the clinic that patients that secrete high levels of these tumor extracellular vesicles are less responsive to immunotherapies, and we have exciting data showing that this may be the case. So essentially what we're looking at here is this is the end of a mouse experiment. So this is a tumor mouse study. We inject nanoparticles into the tumor or into the mouse, and then we take out the organs and see, did they go to the tumor or did they go to healthy organs? And we always face this challenge shown here. So we have different organs that we're listing here on the side. These are mice that have a colon cancer tumor. This is the classic challenge that we face in nanomedicine. The majority of these nanoparticles accumulate in the liver. The liver is a sponge for nanoparticles, essentially, and we're getting inefficient delivery of the lipid nanoparticle into the the tumors. But what we did, this was led by a very talented postdoc, Ning-Chang Gong. He's now a professor at USTC in China, in collaboration with an amazing exosome biologist at Penn, Wei Guo. We then took that same tumor model, but using CRISPR-Cas9 technology, we edited out a gene, Rab27A, to stop the tumor from secreting these extracellular vesicles. And we saw the striking result that when you prevent the tumor from secreting these vesicles, less of the nanoparticle is going to the liver in this case, and we can now get better delivery of the LMPs into the tumor. If we look at these interactions under cryo-EM microscopy, we do see that these LMPs and these extracellular vesicles derived from the tumors do interact in this case. So what is going on here? I won't go into all of this. This is very complicated flow cytometry. We did many of these different panels confirming where these nanoparticles go. But essentially what is happening is shown here in this schematic below. This was drawn by Joy Wolfram. She did a nice Nature Materials news and views on our article. And essentially what's happening is when we're delivering the mRNA nanoparticle into the bloodstream with our cancer therapeutic, it's delivered systemically. But what happens is it comes into contact with these extracellular vesicles. The EVs will absorb to the LMP. And what happens is when they get absorbed to the LMP, they'll shuttle away from the tumor. And then through molecular interactions with the EV and macrophages in the liver, they get shuttled into the liver, and then these nanoparticles get destructed. So it's almost a natural clearance mechanism that the tumor is developing to prevent accumulation of foreign particles. So this was a very interesting biological finding that we made, but the question is, well, can we potentially leverage this in any way therapeutically? Because of course, we can't take out the tumor, CRISPR-Cas9 gene edit it, and then put it back in. So what we did instead, this was in collaboration with Drew Weissman's lab at Penn, was we developed an LNP approach that delivers two RNA drugs. It delivers mRNA, which has the immunotherapy, and it delivers siRNA to shut down transiently the secretion of these extracellular vesicles. And here are the results of this in vivo study. So we have three groups that we're looking at here. In pink, we have the siRNA that shuts down the EVs, the biological barrier, no RNA drug. In blue, we have the mRNA drug, but no siRNA to shut down EV secretion. And then in red, we combine the immunotherapy with the EV approach. We see here, if we just shut down the EVs secreted from the tumor, it has no therapeutic effect. The mice tumors grow, similar to controls. They all die within 20 days. If we deliver the mRNA therapeutic here, this is an mRNA encoding for STING, which is incredibly toxic if delivered off-site, but if we could deliver it precisely into tumors, it could turn a cold tumor into a hot tumor that can respond to immunotherapy. We do see that we could reduce tumor burden somewhat, but these mice ultimately die after 30 days. But if we now shut down the EV secretion so that more of the mRNA drug can get into the solid tumor, we can now deliver STING in a very potent manner. And through systemic administration now over a 50-day period, we could see 100% cures in these mice. So going back to my original analogy, so we develop LMPs for RNA delivery to deliver drugs, but if we also use LMPs to shut down biological barriers, we can now improve systemic delivery of mRNA drugs into solid tumors to now go after the holy grail, which is getting the systemic delivery of RNA into these solid tumors. If I have time, I'll briefly mention some of our work in fetal and maternal health as well. So we're also very interested in using mRNA LNP technology to treat deadly pregnancy disorders. The reason this project came about is when the COVID-19 vaccines came out in 2020, we actually received many emails from pregnant mothers asking, I'm thinking about taking the COVID-19 vaccine. Is it safe? And of course, the challenge is many pregnant mothers are excluded from these clinical trials. So of course, I'm not a clinician. We know it's safe. But from a drug delivery perspective, we did not have a great understanding of, in pregnant mothers, is the distribution of the mRNA LNP vaccines changing? So this led to a very exciting project where we explored the placenta as a therapeutic target for mRNA delivery. Of course, the placenta mediates nutrient and oxygen transport between the mother and the fetus. It's exciting from a drug delivery perspective because up to 25% of the cardiac output supplies blood flow to the uterus and the placenta. And there's also a range of disorders that we can treat if we can reach these cells and tissues. One of the major ones, of course, being preeclampsia, which could affect up to 8% of all pregnancies. And there's non-curative options to treat these patients. So I won't go into all of the results behind this, but basically what we did was we went on a fishing expedition. So this was led by Kelsey Swingle, a PhD student. She's now a professor at Rice University, to identify next generation LNP targets for RNA delivery. She found ones, I don't have time to get into it, but she found ones that don't cross into the fetus, but paint the placenta and transfect a variety of cells. And she asked the question, well, now that we hit these cells, can we deliver therapeutics such as VEGF to restore the vasculature in the placenta and have healthy nutrient and oxygen transport into the fetus? And I'll show one result here that was quite striking that we did. So we basically had pregnant mice. They gave birth. We see in the PBS control, these fetuses are healthy. In the disease model with LPS, you're not getting good nutrient and transport of oxygen into the fetus. These fetuses are shriveled up. But in the disease model, if we now deliver the LNP to deliver VEGF and restore the vasculature, we see the result is quite striking. These fetuses look almost identical to the healthy controls in this case. And we see this as a new mRNA platform going forward for a range of placenta and pregnancy disorders. I'll end on a quick note that we're very excited about translating these technologies into the clinic. One of the first technologies that I mentioned that was led by my PhD student, Maggie Billingsley, she's now a postdoc at MIT, focused on this concept of in vivo CAR T-cell engineering, and together with Drew Weissman, Carl June, and others, we started this company known as Capstan Therapeutics that originally started on cancer immunotherapy, but then we found that this CAR T approach was very potent for treating autoimmune disorders and treating autoreactive B cells. This is now in the clinic, phase 1 clinical trial in Australia. It was recently acquired by AbbVie, and we're now developing a next generation company, Liberate Bio, that can now target the innate immune system, such as macrophages, and reprogramming them to target solid tumors. So in conclusion, there's many opportunities for mRNA therapeutics beyond vaccines, but as we've shown throughout the presentation, they'll require new delivery technologies to reach these cell and tissue targets. And we went over a variety of examples for cancer immunotherapy and also our work in women and maternal health. I thanked everyone throughout the presentation, many amazing postdocs and grad students, and also our collaborators, Drew Weissman, Carl, and Wei Guo at Penn. Thank you so much. I would be happy to take any questions.
Can we start here in the middle? Lisa?
Lisa Volpatti, Northwestern. Thank you, Mike. Great talk. Really impressive, all the work that you've done. I'm sure everyone agrees with me there. So my question is, you showed like the targeting of your LNPs, but I'm wondering if you've looked into where the cytokine or growth factor goes that is expressed as a result of the mRNA. And if that kind of gets leaky or goes everywhere, or if it stays locally to the tumor or the placenta, depending on the application.
Yeah, it's a great question. So I'll start with the placenta example. So originally when we started the placenta project, the common thought in the field of mRNA delivery is if you want to do a protein replacement therapy, we'll use the liver, which is of course our natural bioreactor in the body to produce therapeutic proteins, and leverage that to secrete VEGF. And we saw this interesting result where the liver, if we use a liver-tropic LNP and we delivered it, it delivered an order of magnitude greater VEGF into the bloodstream compared to our placenta-tropic LNP. So compared to the liver-tropic LNP, our placenta-tropic LNP, in terms of overall secretion, was much worse. But in terms of vascular normalization, the liver-tropic LNP had no effect on the vascular normalization, But with the placenta-tropic LNP, when you secrete that protein locally, it induced vasodilation. So what we think is happening there is when we deliver it systemically, it has a short half-life, it's getting degraded, it's going to off-target tissues. But when you develop an LNP where you get local secretion and you have that protein in its most bioactive form, that's ultimately the secret sauce in this case. And then going to your other question with IL-12, that was an intratumoral example. So IL-12, I will mention, a challenge with IL-12 delivery is if you do deliver it systemically and it goes off target, it can have severe toxic side effects. So what we're able to see with our tumor-tropic lipids in this case that we deliver directly into the tumor tissue, we get high levels of uptake of those LNPs and we get minimal. We still get IL-12 that's coming out, but it's at orders of magnitude less than if we took a systemic delivery approach. And we've really seen minimal toxic effects. And one thing I'll mention as well, another company, Strand Therapeutics in Boston, they're developing an IL-12 intratumoral therapy, and they've seen a very favorable safety profile as well with localized IL-12. So we do think there's a path forward here into the clinic with the intratumoral approach.
Thank you.
Chad, no. There was a question here. While you're handing the mic, what's the maximum size of the cargo that you can deliver? If you're thinking about trying to, put several proteins in, how much RNA can you pack in these particles?
Oh, that's a great question. I could go all day about that. So if we think about if we compare it, non-viral LNP technology to an AAV, so a viral-based therapy, a challenge that we've faced for decades is they have payload limitations. So if we want to secrete very large therapeutic proteins, we can't do that with a viral system. But the benefit of an LNP approach is there's, in theory, no limitation to the size of the RNA cargo. As you have RNA cargo that's larger, your LNP will become larger in diameter. It could be more difficult to deliver systemically, but in theory, there's nothing stopping you there. The challenge becomes if you're delivering multiple different RNA constructs or DNA constructs, I would say, in the field. So if we're delivering gene editing constructs, that requires an mRNA, what's known as a guide RNA, and potentially a DNA. When you deliver all three of those within an LNP, as we've heard, Chad's done great work on this. We had a great talk about this last night. LNPs are not homogeneous. They're very heterogeneous. When you try to encapsulate multiple payloads, you might have a subfraction that have all three, but then you might have some that just have one or the other in it. So it becomes challenging when you're delivering multiple different constructs versus just one larger payload. It's a great question.
Thank you.
Okay, when you're delivering the LMPs to tumors to prevent the growth of vessels in the tumors, is there a difference between the requirements of different types of tumors?
That's a great question. So for the tumor study in this case, we weren't targeting the vessels in this case. We were actually targeting what we call the overall microenvironment. And in the case of the IL-12 study, we wanted to create an environment in the solid tumor that was less immunosuppressive, that could be responsive to immunotherapy. But that's a great question because there are some tumors that will respond well to immunotherapy. We started with the MC38 model because it's known to be immunogenic. That's the first model you test for immunotherapy. But there are some tumors, solid tumors, that are very silent and immunologically cold that are harder to treat. So in the next phase of this study, one of the harder ones to treat is metastatic breast cancer. It's very hard to treat with immunotherapy. Also, late-stage prostate cancer is very hard to treat. It's not responsive to immunotherapy. So we're now moving this forward into these solid tumor models that are less responsive to immunotherapy because, as you said, it's very heterogeneous. It's going to be different from tumor type to tumor type.
Okay, on the right side.
Hi, thanks again for that talk. I think the work that you've done in the high throughput discovery is super, super cool. I'm curious though, has there been any design rules or like structural features of the lipids that you've identified as being tropic to a particular tissue? Are there any similarities in those structures that you can now predict what lipids actually will go where?
Yeah, no, that's a great question. I think this is going to be a major area for artificial intelligence. going forward. So we're by no means an AI expert, but when we give this talk, many people who typically attend these talks who do work in AI, one example I'll bring up is my colleague Dan Rieker. He's in Duke Biomedical Engineering. He mentioned that, with the large library approach and the amount of data that we're generating, this is great for developing design rules and coming up with new structures. And there's certainly the case. It's hard to say one structural motif is never going to be a design rule for everything. So if we talk about, say, the amine head group, if we have an ether in it, is it going to hit, are all of these lipids going to hit immune cells? No, but we've generally noticed if you tweak the amine head group and you make it more hydrophilic in nature, you could get more immune cell delivery versus liver delivery. If we create, lipids have branches, if we create larger branches versus if we think of a tree, smaller branches coming off the branches, we can improve delivery to, say, the placenta and other organs. So we've made many of these different observations. It's hard to be entirely generalizable, but we're seeing some interesting results, hopefully presented at a future symposium where AI can actually take this data and develop better structures. AI can't make the structures from scratch. You still need the great idea behind the chemistry and the nanoparticle delivery system, but if you feed it the data set, it could further optimize the system.
Great. Other questions?
Thank you for the great talk. I have two questions. So the first one is, how did you characterize the encapsulation of the mRNA? So how did you figure out whether it's actually encapsulated or kind of stick around the surface of the nanoparticles? And the second one is you mentioned about the traditional problem of the nanomedicine. So do you think not just the tumor case, but all the other nanomedicines are going through the same mechanism through the EV that helps it feed more to the liver?
It's a good question, right? I don't know if I can answer entirely within a minute, but when we talk about the encapsulation, so what we do with encapsulation is very standard ribogreen assay. So with the ribogreen assay, essentially you're cracking open the LNP like a walnut, and then you're dyeing the RNA, and then you're getting a signal out of it. There's also other assays as well where you could have a membrane impermeable dye that will only label any RNA that's on the surface. But in most cases with LNPs, because of the nature of RNA being hydrophilic and negatively charged, you really don't see any RNA that's sticking out of the particle. The question about EVs is an interesting one. We have a variety of projects now looking at these interactions. We're excited about them because, as we know in the nanomedicine field, when nanoparticles generally go into the bloodstream, proteins will absorb and form a protein corona. EVs, very similarly, will have proteins on their surface and absorb to LNPs. And in some cases, this could hinder delivery, like we saw in the solid tumor example. But we actually have exciting data now showing for a vaccine that certain types of EVs could actually improve delivery into lymph nodes to create a more potent vaccine in elderly patients that don't respond to it. So we think there's a lot there, potentially leveraging delivery to different organs, both hindering delivery, but we could also potentially exploit it for better delivery as well.
Thank you so much.
Okay, this has been incredible. Let's thank Professor Mitchell and all the speakers this morning. Okay, that brings us to the end of this morning's session. It's a break time for lunch on your own. We'll be back at 1:35 for the afternoon. We've got a couple great talks and a panel where we'll hear from the speakers on some really interesting questions for all of us, the students too. And let me just ask the speakers and session chairs to come up front for pictures.